how's life?
How should it be?
Better than this, I would think, but what can you do?
Yeah.
Dan, Felix, how are you guys?
We're good. Life is better than, life is good. It's much better than the alternative, so.
Agreed.
Yeah, it's always hard to find a good answer to that question these days.
Yeah. I hear they're increasing.
Yeah. State of emergency.
Mm-hmm. How are you doing, Norm? Haven't seen you
Great. I'm sort of glass half full on this thing. First of all, family's well, and there are silver linings in this particular situation, and have been enjoying taking full advantage of them. So it's not been a terrible year.
I'm glad your glass is half full.
Yeah, maybe even 3/4.
Last time I think we spoke, it was more than that.
Slowly going down.
Or a new glass. So just before we start, we're just killing time. I dislike personally these before the introduction, before the start couple of minutes, because you don't know what to do with yourself and it's awkward not to talk. But I wanted to thank everyone for coming and joining. It's exciting to talk about this stuff as a diversion from the 13 hours of other Zoom meetings that precede this.
Thank you. Thanks for having me.
Well, I think we're going to be judging posters again this year at surgery.
I saw that. Yeah. I always do it. So.
Giving back, you know. So we have Yasmin did a fabulous job. We have about 190 people plus registered. Apparently, animal models are popular.
Sure.
So Norm, it's 5:05. You ready to go?
Ready.
Perfect. All right. So
You'll begin?
Yes, I'll do a quick intro to everyone. So hello, everyone. Thank you for joining us for our January TR Talk session. As you may know, our topic for today is the trouble with models, cells, animals, and simulations. I wanted to start by thanking the Translational Research Program, Medical Innovation Toronto, and the Health Innovation Hub for providing us with this talk. I also wanted to thank our panelists for joining us. Normally, I would do the honors of doing the speeches, but today our moderator Norm is going to take over that for me, which I appreciate. So for further ado, Norm.
Well, good evening, everyone, and welcome to TR Talks for today. And it's a real pleasure for me to serve as your moderator on a topic that seems to pique a lot of interest from many investigators and other individuals involved in health research. Today, we have three terrific panelists who are going to address this issue. And let me begin with some introductions. I am a pediatric nephrologist and physician scientist at the Hospital for Sick Children and a scientific director at the Canadian Institutes for Health Research. I am a biomedical researcher. I've been very involved in translational research at the level of certainly the education of young investigators, as well as some of my own work. And this is an area that has particularly interested me, since a great deal of my career has interfaced with the use of experimental models to try to model and to understand human diseases. Let me go to our panelists. I've asked each of them to introduce themselves in terms of their academic positions and their research. and how they interface with this particular topic, all in about 60 seconds or so. And so let me begin with Margarete Akens, who please introduce yourself to the audience.
Thank you very much, Norm. So my background is in veterinary medicine. I studied in Germany, and then I was trained as an equine veterinarian. And during the time I did my dissertation, and I got so much interested in research that I did a PhD in Switzerland, also at the vet school there. And then I came to Canada as a postdoctoral fellow for 2 years, which I spent in Guelph. And during my PhD and my first postdoctoral fellowship, I worked with sheep. And then when I came to Toronto, I downsized to rats, and I'm now working ever since on a preclinical animal model, mainly of bone metastasis. Using, I have also done some fracture healing in different studies, and also worked with rabbits and pigs as animal models. Oh, and I'm sorry, I forgot to say that I'm a staff scientist at the Techna Institute from the University Health Network and an assistant professor of the Department of Medical Biophysics and Surgery.
Thanks, Marguerite. Dan Drucker.
Hi, everyone. I'm a clinician scientist at the Lunenfeld. I occasionally say this is crazy stressful times for everyone, especially for trainees. So the university and the hospitals and the faculty have great resources to help people who are stressed out. I have my own personal department of psychiatry in terms of one of my daughter-in-laws. But if you guys need help, call us, email us, text us, we can help you. What do? I work at the Lunenfeld. I'm an endocrinologist, and I make my living studying mouse models of metabolic disease, diabetes, obesity, gastrointestinal disorders. But I happen to be very critical of our enthusiasm for extrapolating a great deal of what we do, sometimes uncritically, to humans. And perhaps that's why I'm invited to speak here. I also am a professional troll on Twitter, where I comment on papers, trying not to offend too many people, but sometimes unsuccessfully.
Thanks, Dan. Felix.
I'm Felix Ratjen. I'm also at the Hospital for Sick Children, and I'm a senior scientist at the Research Institute. Senior is not because I have gray hair, but it's a career step in the institute. I'm also head of pediatric respirology, and I'm a pediatric respirologist by training. I also co-lead the CF Center, and one of the goals of the CF Center here is to build models that we can use to predict treatment response, and that involves patient-derived materials, amongst others. And I am more on the clinical trial side, and so how we can use compounds to move them forward into clinical trials through models. And I'm also head of translational medicine, which is a program within our research institute. So, and as you may hear from my accent, I'm also originally from Germany.
Well, thanks, Felix, and thanks, everybody. Let me just say a few words about the format for today's session and how all of you who are participating can feed forward your questions and comments. We have the panelists and I have agreed on some questions that they're going to address sequentially, and I'll moderate that. And while this is happening, no doubt you may have, you in the audience may have questions that you wish to send to us, and you can use your chat function to do that. Meanwhile, we'll ask you to keep your speakers on mute and your video off to preserve bandwidth. And Yasmin is going to keep track of the comments and questions that are put forward, and she'll feed those forward to me. And we'll try to represent the major questions that come forward in the discussion period. So with that said, and I hope that's clear, let me begin with the first round of discussion by our panelists. And as you could see from the title for this TR talk, it was the trouble with models of various types. So the question really is there really trouble with the nature of the models that are being used, or is there trouble in the way that they're being used, or do we have unreasonable expectations of the models that are being constructed by investigator groups? Of course, all of these things might be true, and I've asked each of the panelists to spend a little time reflecting on this particular question. So perhaps, Felix, would you like to begin?
Sure, I'm happy to begin. So I think the answer to the question is probably there's a component of all of the above. So there's certainly, we can't have the expectation that one model will give us all the answers and will be a perfect prediction for how we can utilize that in translating that into what to expect in humans. So I give you an example. So in CF research, of course, people have tried to use animal models to predict, to use them as models for the disease. And the typical way you go, that you start with a mouse model and to do a knockout mouse that doesn't express the protein, in this case CFTR, and that was relatively easy to do. But then one of the tricky things was that when this model was developed, and it actually did not show the typical manifestations of what we see clinically, which is lung disease. And these mice, despite the fact that they don't have CFTR expression at all, do not develop lung disease because there are other channels that compensate for this specific abnormality. But there are other aspects that have been quite helpful. These mice have intestinal disease, and the intestinal disease manifestations can be nicely pursued and looked at in this animal model. So it really depends on, so models might have specific utilization in some areas, and they may not be so good for other areas. But on the other hand, there's certainly a great interest in using models in different ways. And one of the ways which is very close to the clinic, that actually if the FDA, the Food and Drug Administration, has accepted models to extend the label for some of the drugs that treat the underlying defect in cystic fibrosis using CFTR modulators, so drugs that specifically work on the underlying problem. And interestingly enough, the models that have been utilized and which have been accepted are not necessarily human cells, but FRT cells, so Fisher rat thyroid cells, in which a specific mutation is expressed. And then if you can see that you have a functional improvement with giving these drugs, that has been sufficient for the FDA to accept extension of the label for these drugs. So while I'm not on board with this specific example, because I actually think that one should use human-derived cells, I think there's certainly lots of applications that we can look at in terms of using these models without the expectation that each of the models will give us all of the answers. So we have a program that we call CFIT program, which stands for Cystic Fibrosis Individualized Therapies. And in this program, we are trying to use human-derived models, epithelial cells and stem cell-derived models to predict what the treatment response to therapies is. And what we learn is that there's quite a bit of variability between the different models. And in the end, we will probably not be in the situation that we can say one of these models will give us the answers for everything. And it might be a combination of different models that will help us to really find the answers. So I think I might stop at this point and let others chime in, and then we can start a discussion about this.
Okay, thanks, Felix. Dan, your thoughts?
Well, I'll take a different tack. I think what we all need to do is sell our science. At the end of the day, we're always selling our science to the granting agencies, so they're going to fund us. Certainly to the journals, so they will publish what we wrote. But we also have to own what we publish and own what we write. And so we want to be able to look back 3 years, 5 years, 10 years later, and basically not have someone say, did you really write that? We're like, you serious that you made that claim, that extravagant, over the top claim about your science? And so I think we need to walk a fine balance. And so, I'll use examples from my area. The best model for type 1 diabetes that we have is the NOD mouse. And every week there are 3 to 5 papers published claiming to be able to prevent or sometimes reverse type 1 diabetes in NOD mice, which pretty much those claims have a 99.9% predictive value of failure because they don't recapitulate in humans. And I can make the same claim about brown adipose tissue, amazing preclinical data for the treatment of obesity and diabetes. Humans generally don't have much brown adipose tissue. 95% of men have no brown adipose tissue lighting up on PET scans, and there are no BAT therapeutics in the clinic. And I can make the same sort of discussion about the microbiome in terms of metabolic disease and diabetes and obesity. Manipulating the microbiome is amazingly powerful in animal models of diabetes and obesity. Basically has the smallest effect size or no effect size in humans when you do FMT. So I think we need to love our science, we need to love our animal models, and we need to sell our science, but you don't want to look back at your work and what you've published and the claims that you made and said, oh my gosh, I have just constructed this massive house of sand that has no credibility and no translational relevance. And so that's not an easy thing to do. That requires a lot of thought and just the right amount of hubris and enthusiasm to get your grant, to get your paper published, but not so much that you've spilled over into the give me a break, that's never going to work side. And that's something that we think about every day when we do our science and we publish our science and we write our grants and we write our papers. And lots of examples in everyone's field. I'll stop there.
Okay, thanks, Dan. Margarete.
Yeah, so the short answer to all your questions is yes. And it's very important to, first, you can never 100% translate something from an animal model to a human. So it's extremely important to make the right choices. You need to know what you are looking at. Are you looking at treatment effect on a disease? Are you looking for mechanism of diseases? Are you going to develop a device and test it out in an animal model? So you really have to be aware of your question. Then you have to be aware what type of animal model can you use? Is the best model available to you? So there will maybe restriction with the species, with how you are going to take care of them and the cost involved. So that's something you have to consider as well. Then you have to be aware, and Felix and Dan mentioned this, there are differences between mice and humans, and there are also differences between other species within them and also with humans. So you have to be aware of all these differences in anatomy, physiology, and metabolism. And I can give you later some examples. If you work with people who are very focused on one species, they forget about this. Like I had physicians, the one species doctors, asking me, oh, we want to do some work in knees, and the animal has 4 legs. Does it have 4 knees? I say no. Or in other areas where people were using a drug in mice, and then they were using the same drug in rats, and they used the same dose, but they killed the rats because the metabolism of this drug was different. So there's a lot of this to consider before you start the experiments. So that is a very important part. And then also, depending on the type of research somebody is doing, it should be then tested also in other animal models, or even if it's available, and that's something I'm interested in, but it's going a little bit slowly, is if you would use natural occurring diseases. So the other big problem is that a lot of the animals being used are genetically modified, they're specifically bred, so that they, even if they get some diseases or you induce them, they're not really giving the natural occurring spectrum. So if you say, okay, I don't want to jump into human right away, so I will look at animals with diabetes, or I look with animals which have the same type of cancer. And there is a lot of research going on where you can see what the differences are in these diseases between the different species. And then you can use that as a way to further evaluate your treatment or theory before you go into humans.
Thanks, Margarete. So just before we go on any further, any of you would like to comment on anything you heard from the other speakers at this point? Any of the points raised deserve further comment in your view?
We'll just keep going because we can talk forever on this.
So one question that I'd like you to comment on at this point is obviously the entire enterprise from a translational medicine perspective is to develop insights, mechanistic insights, pathogenic knowledge, and solutions that relate to human beings. So we're focusing on the utility of models, but what is the burden for translational researchers, either in partnership with others or themselves, in defining human physiology and pathophysiology so we have a gold standard for what these models are supposed to represent? What are your thoughts about that?
Pick someone. You want me to start? So
Sure, Dan.
I think some diseases are really easy. So in my field, diabetes is type 2 diabetes, very easy, lots of models. Obesity, very easy, lots of models. NAFLD, NASH, very easy, lots of models. Then you get in other fields. So I mentioned type 1 diabetes, impossible because we don't understand the disease, and the positive predictive value of the gold standard model, NOD mice, is 0. Or we get into neurodegenerative disease, where there are dozens and constantly new models for Alzheimer's disease, neurodegeneration, and the positive predictive value for those models is 0% as well. So some fields are unbelievably difficult because there is yet to be a really good validated model, and other fields like the ones I work in are simple. And I think it just really depends on the field and the disease that you're, sort of going into, that you have either 0 barriers to use of animals, or it may be fundamentally extremely difficult to gain acceptance or for what the best models might be.
But Dan, I think, and everyone, the point I was trying to make, and excuse me if I wasn't clear, is I was really asking about the interrogation of human disease, the adequate interrogation of human disease, in order to define the parameters that really do need to be modeled in animals. It's clear that the NOD story, the neuroprotective story, might be has been considered by many people to be a complete disaster because they are not predictive of what's going on in human beings, yet they are persistently used. There are other examples of human asthma, pneumometavirus, et cetera, et cetera. My question is there enough human interrogation going on such that we have enough reference points for setting up models that would serve the preclinical model?
I think for some diseases, the answer is yeah. We know what FDA approvable endpoints are for cystic fibrosis, for acute renal failure, for type 1 diabetes, for Alzheimer's disease, etc. And sometimes you can model those effectively in animals, and other times it's almost impossible. And you're using surrogates that are not relevant. So I think, a good place to start if you're really interested in translation is to say, is there FDA guidance on what it would take to get this drug approved? And what are those endpoints? And can I actually measure those endpoints in animals, or maybe they don't even exist as endpoints in animals?
Yeah, maybe I can comment on that. So I think in many areas, there are multiple components that are relevant in the disease process, and we don't always understand which one are the main features that are relevant. And in that sense, animal models can be somewhat helpful, but it often has to go back and forth in terms of understanding some of the components that you can then model and then go back and look at them in context. So for instance, in the field of CF that I'm working with, so some of the aspects such as mucociliary clearance problems are easier to model, versus infection is much more difficult to model because of the differences in developing infections in animals, and also the different pathogens that animals have compared to humans. So it becomes complex because if you break down and try to isolate one of these components and study them, of course, then to really understand them in their relevance, you have to go back and forth, because you won't be able to have one model that will perfectly define all of the aspects. So it is sometimes quite complex, and certainly in the field of CF, this has been going on for decades now, and there's still significant components that are not easily understood, such as whether inflammation is a primary event or a secondary event. We have been discussing with that for 30 years, and you should be able to model that relatively easy, but it has not been easy, neither in epithelial cell models and in animal models. It's been really complicated to get at the chicken and egg question for some of these aspects.
Margarete, anything to add?
Yes, so I think it's like I had mentioned before, it's very important to know what question you want to answer. And also it's to know more about the physiology and metabolism of the specific animal you choose for your model. If you think, okay, I have 1, 2, 3, 4, which is present in the human disease, now I have to put that in my rat or my mouse or whatever, it may not be possible because they are missing some factors, or they are metabolizing things differently. So it's much better to say, okay, this is how it goes in human. Is there any animal around which has a similar disease or a similar mechanism? Because there are, like, I'm mainly working in the musculoskeletal area, so I'm not familiar with a lot of the other diseases, but there are people who started studying influenza on ferrets or other respiratory diseases, because it's a much more comparable model than using mice. And what I also notice very often, people stick to the species they are used to. So I have heard, when I ask people, I say, why did you studied this disease in young mice? Oh, we always use them. That is not the right answer. Like, I have seen papers where people publish work about treatment of osteoporosis, and the model they used were young male rats. And I think it's pretty much known that osteoporosis is a disease in the older female. So a lot of people, they just do experiments with what they're used to, and they don't look outside of the box to find ways to get to where they want to be. And like I say, it also depends. Like, we use the bone cancer models, and obviously the way I introduce the bone metastasis is not the way it happens in people. But what we want to have a certain picture to be able to analyze treatment effects, to see the changes in the bone, and that is what we get. We get what we want to get the answers to our questions. And we were also successful in using the preclinical results to transfer it into a phase 1 clinical trial. But yeah, like I said, it's very important to make sure to think outside of the box and don't stick what you know to what you know.
Great.
And there was something
Yes,
which becomes much more important is the reproducibility of experiments. A lot of journals now require that you adhere to the ARRIVE guidelines. That means to the animal research reporting in vivo experiments. In the past, there were the three R's to follow: replacement, reduction, and refinement. And now they added a fourth R, which is the reproducibility. So that not some lab published tons of data on the treatment or pathology of one disease and nobody else can reproduce it. So that's where the journals now put also much more effort. So that's also an important part.
Yeah, I think you're drawing attention to two separate categories in this discussion. One is the face validity of the animal models themselves and their relationship to the human condition. And then there's a huge issue around methodology. For example, people in industry have provocatively written in such journals as Nature that for generally when they read academic papers using an animal model, they often take it with a grain of salt or not at all, and feel that the whole thing needs to be reproduced in much larger numbers and with much greater methodologic precision, like you would run a human clinical trial in order to believe any of the results that emerge. And this is, I think, related to the last point you were making.
Yeah,
Margarete.
and it's also what I noticed is when I was looking, I was treating one group with zoledronic acid, it's a bisphosphonate, a drug which is being used to treat bone cancer patient to reduce the further resorption of the bone in the cancer. And when I was looking for the dose to give to my rats to mimic the human dose, I found a lot of papers which had used 3 times as much, and they injected it 3 times a week. They got very nice results, but it didn't mimic the dose you would give to a patient who suffers bone metastasis. So then what I did to to find the right dose for my rats in the experiment to mimic the human condition, I just contact Novartis and I said, what is the rat equivalent dose for what you would give to humans? And they just provided me with the dose, and we gave it to the rat once every 3 weeks. And that mimics the same a human would get, and not the 3 times a week, 10 times the dose, just because the animal would tolerate it, and you get nicer results than with a lower dose.
So let me ask you all to comment on the following. I think it's clear that the complexity and the specificity of the human condition as relates to a whole set of variables, life course issues, we just heard about that, genetics, genetic background, pathobiology, comorbidities, which are often complex in humans and simplified in animal models, co-treatments, coexisting treatments. These are some of the things that really affect the validity and predictive power of non-human models in translating what we want to humans. So I guess the challenge that I'd like you to comment upon is, given all this, what are your recommendations for improving our current situation? Because I think we have reason to be rather critical of the transferability of what is being learned from a translational perspective, not necessarily a scientific perspective only, but a translational perspective from models of various sorts to human beings. So what are your recommendations based on your knowledge and experience on how our field can improve its track record in this regard? Who would like to comment?
I can start. So I think, Norm, the answer is different for every single area of research. And so, as I mentioned, diabetes and obesity are extremely simple human disorders to model in animals. The endpoints are extremely simple. The drugs work in animals, the drugs work in humans. There's no difficulty whatsoever. And there are lots of different animal models and they all have merits. I would argue a monogenic disease, so you really understand if there's a mutation in that gene, cystic fibrosis is being mentioned, although the mice might not perfectly recapitulate the human disease, you have a tremendous ability using genetic technology to interrogate the pathophysiology of that disease. And then there are just so many diseases, including coronary artery disease, including type 1 diabetes, multiple sclerosis, neurodegenerative disease. I could give you 100 diseases where if you don't understand the human disease, then a great deal of the effort, understandably, is to try and develop new models that might somehow approximate features of that pathophysiology. So it really depends where you're working, what disease you're working on, what the state of translation from humans to animals and back and forth, and it will be different for every single disease. So if you're like me, you're a simple endocrinologist, you pick diseases that are easy to model from humans to animals. If you really want to bite off a lot more, you pick multiple sclerosis, Alzheimer's disease, and type 1 diabetes, because no one has figured out the animal models for those.
And in those situations, Dan, what is your opinion about the repetitive use of models, for example, in type 1 diabetes, given the track record in translation?
Yeah, so people get very offended, and I've offended a lot of people. If you stand up in a meeting and say, seriously, the NOD mouse that's being used in 500 papers, which have all failed when those efforts have been tried. So people will get very offended. And I think there does need to be, I think, an effort to develop new models. So in the neurodegenerative disease space, many of the foundations are funding millions and millions of dollars of just developing new mouse models because they realize that the field is stuck on the conventional double and triple transgenic and tau and senil and stuff. And so I think if you're in one of those fields where the predictive value of the gold standard animal models is almost zero, then you do need to spend a lot of effort with your colleagues at a very high level trying to move the field forward and not just repeating the same old stuff in the same old animal models that have failed to date. So sometimes it's extremely difficult to move the field forward and budge people off the fact that your gold standard model has not been effective in translation. And it gets very emotional and people's careers are invested in using these models, and it's a difficult conversation, but it needs to happen.
Yeah.
I think it's certainly as attractive to use some of that in monogenetic diseases as you mentioned, because it's easier to move research through this platform in a meaningful way, that you go from genetics to molecular biology into some models and then into human derived tissues and finally into humans. It becomes much more complex and questionable for complex diseases, and asthma is one in my field. I mean, there's been a model, the sensitized over mouse that has been used for decades now, and you can pick your favorite cytokine, be it IL-5 or IL-13, which you knock out in the model and you cure asthma. Unfortunately, that's not quite the way it goes in humans, and but it has, one of the reasons it's been such a nice pipeline for people, because you can build a nice career on that and you can publish a lot of nice papers, but it doesn't necessarily mean that it translates into something useful. And I think that's something that we always have to keep in mind and always ask this question, is not just, is that experimentally sound as a model, but does it actually mean anything for the disease process overall? And I think this question isn't always asked sufficiently.
Margarete?
Yeah, so I agree with what has been said. There is only, although I would say there is a way to finally change the attitude of the people in not repeating experiments which don't work. This requires a strong group of people in the research field to speak out about it. I'm also a member of the preclinical group within the Orthopedic Research Society, and we try to get a consensus together in how animal experiments should be performed for musculoskeletal diseases, so that we establish a guideline. Is like, okay, if you want to do a fracture model, this is good for, this model is good for answering this question, this model is good for answering these questions, but go away from model XYZ because it has been proven not to mimic the real life. And if every society or every group would form this committee and work on what is the best model for our disease, or how do we get that, that would be a big point forward. And then the people in Dan's case would stop using this mouse, which doesn't work. That would be my suggestion.
Yeah, so I think, Norm, you know, we fall in love and get very comfortable with our familiar models. And, you know, so in my field in diabetes, widely used models are the DB mouse and the OB mouse. And these are mice that either have leptin receptor mutations and or leptin gene mutations. And you see repeatedly written or demands from reviewers that you should use these well-characterized, excellent models of type 2 diabetes. And if you would actually say to the reviewer, well, you know, there's like 75 people in the world with an OB mutation and maybe 150 people in the world with a leptin receptor mutation. And depending on the genetic background, these humans don't even get diabetes. These are terrible models for type 2 diabetes, yet there are thousands of papers that utilize these models. And every field has this sort of issue where people have just fallen in love and gotten very comfortable, and they know how to use these models to produce their papers and get their grants. And they're very reluctant to kind of look in the mirror and say, actually, this is a terrible model for the disease and for translation. So I think Margaret makes a good point. If every field would have a consensus blueprint on saying, these are the best accepted models in the field, and, you know, class A, grade A evidence, and then these are the ones we should think about moving away from. And these are the ones that are actually not informative at all. And you really don't see that consistently. Some fields have that, other fields don't.
Yeah, great. Yeah, thanks both. Excellent points being made. Let me ask you all, you know, people really wonder about emerging technologies or well-established technologies now. Certainly in genetics, we have CRISPR technology, we have new stem cell biologies, stem cell techniques that are built, that are used in concert or separately from xenografting. We have organs on a chip to go completely in vitro. What are your comments about the emerging nature of these technologies and overcoming some of the limitations in either cells, tissues, or whole animals?
Yeah, so I'll start. So I think depending on what you're studying, they can be like 100% amazing or next to useless. And so, you know, if I'm studying the human liver, the ability to generate human hepatocyte-like cells reproducibly from organoids is just unbelievable and amazing and an invaluable tool. If I'm studying how the gut controls the brain and the pancreas, and I make gut organoids, there's no lymphoid immune system, there's no blood supply, there's no neural innervation, and there's no communication with other organs, and it's basically almost useless. And so the technology really depends, again, on what you're studying and how reductionist you can be and how you can recapitulate the cell and organ of interest. And in some cases, it's just amazing. And in other cases, it's not even worth trying.
Other thoughts on that?
Yeah, and certainly for genetic diseases, that is quite potentially quite powerful. And there are lots of examples now where researchers have basically used these cellular models and then moved directly through some bridging, using some animal data just for safety, but not necessarily for studying mechanism or studying efficacy, and moved right into humans. And that has been very successful in a number of areas. I think, but I agree with Dan that for it depends on the complexity of the disease and whether you can actually, whether you have a defined target that is easy to recapitulate in these models, and then it works very nicely. And so I think there's lots of potential in that, but for specific disease processes and not in general for everything.
Margarete, any thoughts?
Yeah, I have to agree. I'm unfortunately working with one of the more difficult tissues, which is bone and used to be cartilage. So that a lot of people try to grow that in culture and recreate it, and it's not an easy task because it's not just the nutrients and the cells. There's also mechanic playing a role in the development and the interaction with the surrounding tissues. So it becomes very difficult. So I agree with what has been said before. It's a helpful tool for certain things and for screening, but it has to be validated properly and not and put in the right place.
Yeah, great. So I think well, we've already spent 45 minutes enjoying this topic. So let's pick some questions from our participants in the audience, because we've had a fair number of questions now. And dovetailing on the last part of our discussion, someone asked, from an ethical perspective, do you think that recombinant animals can be better created to better mimic humans? So humanization of animals, animal tissues, etc. Where do you think this sits in the kinds of tools that should be used? Marguerite, what are your thoughts about that?
So I have personally no practical experience in that, but if it helps to answer the question and reduces the overall use of animals and doesn't induce more complications, then I think it's a valid tool to do that. But yeah, so I don't have any personal experience with that.
Yep. Other comments?
Yeah, I think it's a fantastic, very important, probably underused technology. So, we know there are just so many differences in human cells and lineages and turnover and metabolism, and also the structure of many human proteins and receptors are different than they are in mice and rats. And so, sometimes you absolutely need to humanize a mouse to have a model for human drug development. But at other times, there's in the immune system, for example, the human immune system and hematopoiesis is fundamentally different than it is in the mouse. So I think this is a really important opportunity to get better data that's translationally relevant by, I will call it humanizing as opposed to recombinant. But sometimes it's just engineering one protein to knock in the human protein and really have a good readout. And other times it's, engineering the entire immune system or hematopoietic system. And I think these are terrific endeavors generally.
Felix, anything to add?
Yeah, I think it's certainly provides unique opportunities, but there are also limits to it, right? So because if you humanize a model, it's still not a complete human. There's still components that are reflecting the original animal. So there's a limitation in that as well. And you always have to ask yourself, can you recapitulate that in human tissue without needing a whole body system? But that's certainly different from disease to disease. And so for some specific circumstances, it certainly can be very useful.
Yep. Well, here's a resource question for you. Imagine that, and I'm paraphrasing the question, amplifying it a bit, but imagine you're rather naive in a particular field. So you don't know all the literature. You start reading the literature, you see how conflicted it can be, or apparently biased, or depending on your point of view. Is there any kind of an online tool that one can go to that provides reputable analysis of human states, appropriate animal models that should be used, or at least balanced arguments that one can, that are summarized, that new investigators, naive to certain areas, could access? Do you know of such resources?
So I think, again, every field is going to have either a wonderful resource or nothing, and then everything in between. So, if you look at the neurodegenerative disease field, there are amazing websites that are critically looking and evaluating all of the dozens of mouse models of neurodegenerative disease, and then highlighting the new ones under development with tremendous amounts of detail in addition to the usual review articles, and then other diseases and animal models. I'm sure there's like almost nothing available. Some of the vendors as well, Jax sometimes has a lot of information about mouse models of certain diseases that's very easy to find and searchable. So I think it just depends on the field. And sometimes there's websites beautifully devoted to this exact topic, and in other fields there's nothing.
Yeah.
What I would like to add to this is it also depends, and Dan just mentioned a different laboratory where the animals come from. That is something where people also have to pay a lot of attention, because even so the mouse or rat has the same name, when it comes from a different laboratory, it has different properties. Yeah. I'm also speaking from experience. Like we normally use the rats from Envigo, used to be Harlan, and we couldn't get them, and we ordered them from Charles River, and they had the same name, but they were different. They didn't take the tumor as well. They looked a little bit different. So that is also something people have to be aware of when they study diseases, is the source, not just of the cells, but also of the animals they combine together, depending on the question. And I agree there, the resources depend on the field people study, want to study. Mm-hmm. But it's also good, I make some advertisement, speak to your local vet when you want to start with animal experiments.
Yeah.
I think the reality is the question is very good, and there should be much more of that than is actually available, because for many fields, the honesty is not necessarily out there to describe that if a model hasn't been shown to be very helpful, that this is actually adequately stated in the literature for reasons that we discussed before, that people have their favorite model, and also that often this is a controversial issue. I was part of trying to write guidelines for a path for specific agents in the field of CF, and it was very difficult to be absolutely clear so that somebody going into this field would be able to extract the information out of that, even though for the majority of us it was clear that some of the models are just crap, are just not useful. But so I think the unfortunate reality is this would be very helpful, but is probably not available for the majority of fields.
All right, here, let's go to another question. So here's a great question for people preparing grant applications. So the question is, how do researchers prepare their criteria for success for their animal model experiments, or whatever model experiments, non-human, if the pathobiology in humans is not adequately understood?
So let me take that on, because I think there's really two, we're speaking here a lot on translation. That's the nature of the interest of this group. Not every scientific question has to be translated into humans, right? And so a lot of us do science for the beauty of science, for the excitement of answering a scientific question in a model organism, and it may be an amazing model organism and a fundamental question of how biology works, and if clearly stated, that's a very respectable, often exciting, sort of thing to do. But if you really are always focused on the human condition, then I think you have to acknowledge, and many of our fields don't have really good animal models, and one has to acknowledge the limitations. And I think most reviewers will give you credit for that. You're basically like everyone else in the same boat. So as long as you are acknowledging the limitations, and ideally not just using if you're only using one model and it's not accepted, you're less likely to be successful if you're using 2 or 3 models, and collectively each one of them might have some limitations. So make sure your boat is large enough and steady enough and constructed to withstand a reviewer's critical scrutiny.
Great, good advice. Any other comments on that point? Okay, well, let's continue, because we got lots of questions. So one of our attendees asked about the accuracy of using stem cells, and we already heard something about organoids. So often stem cells are used to generate organoid tissue versus in animal tissue itself. What are your comments on the comparative nature of these? Stem cell derived versus derivative animal tissues.
Yeah, so again, Norm, I think the answer is going to depend on every cell type that you're deriving from the stem cells, on every organ that you're deriving, if it's an organoid. And in most fields, these stem cell derived cells and or organoids resemble, but do not 100% recapitulate the fully mature differentiated human cell and organ. And in some cases, they're 99%, phenocopying the cell and the tissue, and it's good to go, and the differences are trivial. And in other cases, the small differences are fundamental to the success of the entire experiment and the interpretation of the data. So one just has to be critical. The more cell models that we utilize, whether it's primary cultures or moralized cell lines, iPS-derived cells and organoids, bringing all of these to bear on answering the question strengthens the result of the inquiry, but none of these by themselves is 100% perfect, as we know.
So what I
Yeah,
what I would suggest, like I don't do stem cell research, but if you want to answer the question, if the stem cell generated organoid is matching what is happening in the human, and you cannot do that because you can't take a human experimental model, I would say you have to do all the experiment not with human stem cells, but you have to do that with the species you want to study. So you do work with the stem cells of the specific species, and then you go back into the animal and see how that compares. Then I think it's more likely that you can draw conclusions from doing your human stem cell work to what happens in human. But doing it from human stem cell work and then doing animal experiment in a different species, there is a mismatch, in my opinion.
Thank you.
There's certainly also a lack in terms of the utility of cross-validation of stem cell-based model versus tissue-based models. But for many organs, there's an opportunity to do that. Certainly for the airways, you can compare the models to those directly obtained from humans, and you can do this for the gut to a certain degree by you doing rectal biopsies you get organoids that are derived directly from tissue versus through inducible stem cell approach. And certainly there's quite a bit of that work going on currently, but there's still a lot of unknowns in the equation. And as Dan pointed out, unfortunately, the purity of the stem cell derived tissue is certainly one of the limitations in that process. And so these are all. So this is a complex area, and but I think we will learn more and more as we do these cross validations. Certainly much more difficult for tissues that are less easily accessible in the body.
Mm-hmm. So let me ask you a question again, derived from our audience, and it's a bit different, but I think it's about open science. It's about sharing knowledge and sharing the pluses and minuses of the models that we're using. The question is, does intellectual property play a role in limiting the openness within the scientific community to be sharing appropriate knowledge in order to find ourselves in a different place in terms of how we're utilizing animal models or trying to create new ones for the purpose of translation. What are your thoughts?
I'll start with that. So
Not
I would say
surprised.
no. I would say no. It should have no relevance whatsoever. You know, if you've discovered something amazing, go ahead and file a patent. You have 12 months before that patent publishes and the entire world reads it. In that 12 months, if you want to be secretive and industrious and expand upon the domain of your invention, good for you. But at the end of the day, every one of us on this call wants to distribute our cells, our organoids, our animal models, our discovery reagents, our proteins as widely as we can to have as many people around the world reproduce our data and build upon the data. And that's how we build science. And that's how we become famous. And that's how our work stands tall. So this whole notion that there's a secret and I can't give this to you because I have a patent, etc. I think, sure, for a couple of months, go ahead and develop your invention. But after that, you want this to be widely distributed. You want other people to reproduce your data. You want the field to grow based on your models and cells and findings, not to hide in your own lab and keep it a secret.
Yeah, and the reality for that is it's as you mentioned, it's really relevant very early in the process where people develop their own models. But then afterwards, it's not that you can patent a model and make a ton of money out of it and become famous for the rest of the world. It's really important that the distribution happens so that you can advance the science. So overall, it's probably not the limiting factor in the equation.
Yeah, and as I mentioned earlier, reproducibility is very important and also gains importance, specifically when you read paper. There have been in the past too many papers where only one lab could get the experiments to work and nobody else could reproduce it. So that becomes more important.
Great. So here's another question, again, coming from a certain kind of research and thinking about animal-based research. How can researchers who are using animal models integrate a patient-oriented approach into their thinking and their experimental design?
Okay, so what you do is, and this is my experience, is you don't come as a researcher alone up with an idea and you work on that without talking to other people. Like you talk to physicians if you are not aware of certain, like if you're not a physician yourself and you have a great idea, you make sure that you talk with the physician if that makes sense, what you are thinking. And the same is depending on what you're doing, you involve engineers or other scientists in that project, or pharmacists or pharmacologists, and you involve them very early in the project and plan with them together and do brainstorming to come up with a model, and you see if it's relevant. If you have a great idea or you know somebody with a very rare disease, obviously it's helpful for these few people in the world who may have that, but you want to get something done which helps a bigger population, you have to talk to physicians.
I think every disease is different. And I think for some diseases, what you're really asking the question is, what would success look like at the end of my research efforts if I wanted to make a positive impact for people with this disease? And in some diseases, that's like 100% obvious. It might be, years of survival with cancer, or 15% body weight loss with obesity, or failure to deteriorate in terms of EGFR or FEV1, etc. In other cases, there may not be therapeutics in that area, and you may have to actually build the case for success yourself by talking to patients, by talking to regulatory agencies, by saying, what should we try and achieve? What would change the quality of life? What would be an approvable endpoint? And you have to build that field. So every condition and disease is going to be different. Some very easy, some you're going to be the pathfinder and pioneer.
Yeah, and I think what Margarete was pointing out is this collaboration between the bench scientist and the clinician is very important in this scenario, and bringing them together and to work together and to discuss the questions together is really very helpful. And we're trying to do this in translational medicine in our program more and more, so that we can actually bridge this connection. So when you ask a question, you can actually see whether people on the other side, be it on the bench or on the bedside, can see that this is either a doable experiment or actually a relevant experiment. And doing this early in the process helps to focus research tremendously.
And I just want to add, because we have a lot of different people on the call. So this is really not an MD, PhD thing. So in my field, there are just so many PhDs who completely understand the metabolic diseases, who really understand the patient perspective, who really get the entire picture from the patient perspective. And conversely, I have MD colleagues who really don't get the translational drug development perspective at all. So don't feel bad if you have one degree and not the other degree that you're going to be limited. There's really no limit to your success. It's just how much you want to educate yourself.
And speaking of education, I'm going to just conclude because we've come to the end of our questions. And before I say a few words about MITO, I'm going to first thank all our speakers very much, Dan, Margarete, and Felix. We've had a pretty wide-ranging discussion about issues of face validity and all the attendant issues, reproducibility, and somewhat about the ecosystem that translational research lives in academia and so on. So thank you, all of you, very much for your participation in today's discussion. Let me just say that MITO, for those who are not aware, Medical Innovations Toronto is a translational hub that was created. It's led by Uri Tabori. I'm very happy to participate as well, along with Joseph Fehrenbach, who's on the call today, and the executive of MITO. And it's a coalition of 5 clinical departments within the Faculty of Medicine, allied with the Eureka Institute of Translational Medicine, which is a global community of partners interested in moving the field of translational medicine forward. At the local level, our activities relate to awareness related to translational research and translational medicine, educational activities, and trying to develop a community, a supportive community for researchers in particular, so that they can move their work forward with a translational side to it, and to be supported in doing so. And we have various programs within MITO to do that. What I'd say for those of you who are attending today and are interested in this topic of animal models, MITO is a place where you can put your hand up and say, this is something I'd like to discuss further. I'd like to talk with others about this. And we can facilitate that kind of discussion and are anxious to do so. This panel today is an example of how we try to raise issues that are of common interest and bring people into the conversation. Next week, or is it next week? I'm just looking for the advertisement so I can say this. Yes, next month's TR Talk will be another panel of experts. We'll talk about the front lines of clinical trials and successes, failures, and how clinical trials are evolving in the context of translational medicine. So we certainly invite you to come back and see the advertisement, and I think it'll be a channel just like this one in about a month from now about that TR Talk. Again, thank you, Dan. Thank you, Margarete. Thank you, Felix, for putting the time aside and your excellent comments today. Thank you, Yasmin, for your help in fielding the questions. And with that, I wish everyone a good evening. Stay safe, stay healthy, and take advantage of the wonderful community that we have here at University of Toronto, as Dan started today, to gain support where support could be helpful to you.
And
Thank
thank you to you, Norm, for being our moderator today and making sure this discussion happened.
It's a pleasure to have participated.
And thank you, Yasmin, for organizing everything.
Good night
Thanks,
to all.
everybody.
Thank you all.
Bye.
Okay, bye-bye.
Wow.
Bye.